नमस्कार दोस्तों, वेलकम टू bizbotany। आज हम पढ़ेंगे सीड्स के बारे में। सीड्स, then what are seeds? Seeds are the fertilized ovules. They are fertilized ovules which consist of a seed coat and embryo. They have a seed coat and embryo. Seed coat is nothing but outer covering of a seed, and embryo consists of embryonal axis, cotyledons will be present. In case of monocot plants, one cotyledon will be present. In case of dicot plants, two cotyledons will be present. Cotyledons. On the embryonal axis, you can see a radicle and plumule. Radicle is responsible for the formation of root system in a plant, and plumule of a embryonal axis will responsible for the formation of a shoot system in the embryo. Now we need to understand the structure of dicot seed. Dicot seed consists of a seed coat and embryo. This is a seed coat of a dicot seed which consists of outermost covering. We call it as a seed coat. Seed coat has two layers. The outer layer of a seed coat we are calling it as a testa, and the inner layer of a seed coat we are calling it as a tegman. Inside the seed coat you can see some scare nothing but we call it as a nishana. We are calling it as a hilum, which will help to the attach fruits during the germination period. During germination of seeds, this hilum regions help to attach the germinating seed to the fruit for a food. Above the hilum regions, we have a small opening will be present, small pore will be present. That pore we call it as a micropyle regions. Inside the seed coats, you can see the embryo. Embryo of the dicot seed consists of two cotyledons. Embryonal axis will be present. This region we call it as a embryonal axis. Complete this region we call it embryonal axis. Here, in case of dicot seeds, we have two cotyledons. Two cotyledons will be present in the dicot seeds. They are usually involved in a storing food material. They have a stored food material will be present in these two cotyledons that will be helpful for germinating of the seeds. And on this embryonal axis, this region we call it as a embryonal axis. The upper portion of the embryonal axis we call it as a plumule, and the lower portion of the embryonal axis we call it as a radicles. That upper portion plumule will be helping for the shoot system after the germination of the seeds, and this radical lower portion of a embryonal axis, this part we call it as a embryonal axis. This lower part of the embryonal axis we call it as a radicals that later develops into a root system of the plants. In some seeds, the endosperms are formed by double fertilization. As a result of double fertilization, endosperm will be forming. Such type of seeds are known as endospermic seeds. If the seeds don't have the endosperm, such seeds are known as non-endospermic seeds. Non-endospermic seeds. This is the structure of a dicot seeds, which contains seed coat and embryo. Now we need to understand the structure of a monocot seed. Some monocot seeds are endospermic and some monocot seeds are non-endospermics. Best example for monocot seed is maize. Usually the maize or monocot seed consists of endosperm and embryo regions. The seed coat of the monocot seed, nothing but a special layer. In case of maize, you can see a membranous seed coat which is attached to a fruit wall. This is a membranous seed coat will be present and it is attached to a fruit wall. And it contains a endosperm. Endosperm is somewhat bulky. This region we are calling it as a complete. This region we are calling it as a endosperm region. It is somewhat bulky and stores the food material. This endosperm acting as a storing food materials. Around the endosperms we have one layer. It is made up of proteins which separates this layer. This layer is made up of proteins which is present around the endosperms, which is we call it as a aleurone layers. We separate endosperm from the embryo. This aleurone layer will separate the endosperm from the embryo. Embryo is located at one corner of the endosperms. It is located at the one corner of endosperms, which consists of a dome-shaped cotyledons. We call it as a scutellum. This region, complete regions, we call it as a cotyledon of a monocot seed, and the cotyledon of this maize we call it as a scutellum. Inside the embryo, you can see a small embryonal axis will be present. At both end of the embryonal axis, plumule and radicles will be present. Around these plumule and radicles, some sheath of tissues will be present. Some sheaths will be present. If the sheath which covers the plumule, we call it as a coleoptile, and the sheath which covers the radicle, we call it as a coleorhiza. This is something about the monocot and dicot seeds. Thank you everyone.